to cover basically organic chemistry and biochemistry along with cell biology, all in an hour and 15 minutes or so. It took me a little bit longer to learn that, and of course I've forgotten 95% of what I learned, but I think what I'm really aiming to try to do is, through going through some of the basic points in organic chemistry, give you a sense for the complexity, yet the order, that things we don't ever give any thought to. They're in every single cell, like every single molecule, and we're gonna get into that. So I'll remind you that there's always every kind of science, and really every science has a weakness. Mathematics is probably the closest to being the purest science, but even there you have to examine and keep in mind the assumptions that you begin with. So wrong assumptions and biases, especially in biological kinds of things, can obviously lead to wrong conclusions. And there's a hierarchy of uncertainty. Just because somebody's on TV as an expert giving their opinion, that doesn't mean there's facts or provable theories or principles that are really behind all of that. We've seen that a lot these last couple of years. And as well, things are almost always more complex than they are portrayed, and again, in all aspects of life, but especially even in what I'm going to cover tonight. And I think you're going to conclude, because it's my conclusion, that it takes more faith to believe in evolution than it does to believe in creationism. But yet knowledge is power, so we don't just throw up our hands and give up. We don't need to. We can strive to know what we can know so that we don't have to be intimidated or excluded from these expert commentaries that we hear. And I'm just always going to remind you of John 8:32, and you shall know the truth, and the truth shall make you free. And that a wise man, I don't know why I'm not pointing the pointer, but a wise man is strong, yea, a man of knowledge increaseth strength. And there's 4 principal areas that are sort of proof areas that I'm going to continually reference, or at least some of the evidence will be in these categories. The thing that I think is the strongest is the mathematical improbability, in fact, impossibility of how all these things can come together. At every single one of our sessions, we're going to have 2 different segments today of the second session. And a second is just violations or even misapplications of just pure logic and scientific principles and precedent. And then always understanding the supernatural design versus natural selection to understand how could all of these kinds of things happen. And we're going to see some examples. I don't think that's me with the pointer. Anyway, Lord, it seems to move with me, but I don't know. Anyway, okay. Maybe here's okay. So we're going to see these mathematical improbabilities, and the supernatural design part is what you've heard we mentioned last time, irreducible complexity, how everything has to work from the start together in concert, or else nothing can work. And we're not going to cover as much here with the contradictions of Scripture and theological bases as is conducive for other things. So the purpose of this section is we really want to dig deeper into really the true science, reproducible, known as close to fact as you can be, to understand these myriads and myriads of symptoms of complex systems that have to work together. And even the most basic forms of life share these very, very complex methods. So it's not like I said before, we didn't go from wood burning to coal to gasoline to nuclear energy here. It's like nuclear energy from the start, And we'll show some calculations, and we really want to be challenging ourselves to truly follow the science in this. And I really think most people would agree that this is probably the most injurious area of proof, shall we say, against evolution and for creationism. And there's lots and lots of books, and I just brought a couple that I'm going to make some reference to. Just you may look this Darwin's Black Box by Michael Beebe. He's really the one who advanced most the irreducible complexity. And then this Not by Chance by Dr. Lee Spetner, who is actually, I believe he's a Hebrew guy who was a retired biochemist, and it's always been his hobby. And he's the one who's behind a lot of the calculations that I can share with you. But you'll remember 2 weeks ago, we really left off with the Big Bang. And the Big Bang supposedly had this fomentation of energy that made the elements, and these elements were kind of cooling off somehow and coming together to basic molecules. And tonight we're really going to cover these more complex molecules, the organic molecules. And we're going to really emphasize proteins, which are the combinations of these 20 useful amino acids. Again, a building block that is the order that fits into the complexity that we're going to see. And by the way, I like to throw little numbers in. Most people don't, but I'm a numbers guy. There's 42 million protein molecules in every single one of the 42 trillion cells in your body. So just think of that. The proteins are really what makes everything tick. In the process, we'll talk a little bit about nucleic acids, and we'll make a very quick pass through some of the other organic-type molecules. But the central dogma in biochemistry really is DNA to RNA to proteins. You may have heard if you've taken a basic chemistry, biology class even. And that's really a very trite oversimplification, because what organic chemistry really is it's the chemistry of living beings, of life, of things, and it includes the production of things and functions that have to be carried out. It's energy systems and regulations of all of those things, the functions, the energy systems and the like. And life really involves reproduction. Nothing can be alive if it can't reproduce itself. So what are proteins? Basically, they're just this category of somewhat larger carbon and nitrogen containing molecules that have a very common structure that we're going to go over. And they have many functions. The functions that we know most of proteins are to get cut by eating all this good stuff and working out. But your immune system is made of antibodies. The enzymes that make every process in your body happen are usually proteins. And even in a much more granular level, carrier or messenger molecules, energy transfer mechanisms, and signals such as hormones and other things are all mediated really by proteins. Proteins, this is the basic structure of proteins. We have this basic amine base with a variable piece that gets on it, and that's what makes the amino acid. There's 20 amino acids that are in our body, and what you might want to think of this as is like a charm bracelet. And every single link, which is the backbone of the protein, has an amino acid on it. And it's how that all fits together that really distinguishes the function of a protein. And these are a long chain, and this is, again, a simplification, because you notice that there's polar and nonpolar, charged and uncharged, so that each of these little moieties of amino acids are going to react with each other, and that's going to control the shape of the molecule, which is innately important to the function of that organic molecule. Sequencing this used to be very laborious. Now it can be done in like no time at all. The first protein that was sequenced was insulin back in 1950. Well, at least in 58, Sanger got the Nobel Prize for it. And I don't want to, the Nobel Prize is just kind of a benchmark. It just tells you that these are really seminal, observed things that all the peers would say this is really a monumental task. So I'm not trying to glorify the Nobel Prize winners. Now there's a cute little twist to bigger molecules, and that's something called chirility. And what chirility is like your left hand and your right hand are really the same, but you can't superimpose them. And same thing in molecules, there's a left-handed and a right-handed, we call them stereoisomers. And the interesting thing here, I think at least, is that it's only the left-handed amino acids that can be functional in the body. Similarly, it's only the left-handed nucleotides, it's only the right-handed carbohydrates. And there's a naming convention for why things are left or right-handed. So if you get the wrong one, you can't get the wrong one or it wouldn't work. In fact, it can cause problems. Some of you might be old enough to know or remember about the whole problem with thalidomide. I think it was an anti-nausea medicine given to pregnant mothers in the 1950s or so. And it turns out that the reason that was a problem was that there was too much of a mixture of one stereoisomer versus the other. So, and Pasteur actually is the one we mentioned that last week, who first came up with understanding that. So of these 20, we get 9 of these via our diet, because we can't make it. So it's called essential only because it's essential that we get it from a diet. And there's 11 that our body can make. They're all necessary, but the 11 we can make in our body, we don't have to eat specific vegetables and proteins and things to get them. Now, you might ask, where did the first amino acids come from? Because last time we talked about, well, where did they say the first molecules and subatomic molecules? Well, where did the first amino acids, the basic building block of a protein, come from? Well, a real darling of evolutionary theory is this Miller-Urey experiment from 1952. I think Miller was the student, Urey was the PhD, the lab owner. So it was really Miller who gets credit for this, but What they did is they added certain things to this little reaction vesicle under special conditions of heat and subjected them to electricity. And they were able, to some degree, make a couple of different kinds of amino acids. Now, amino acids can't just float around, they'll die, and not in part because even in this system, there was a lot of other toxins, such as cyanide, that was made. So if cyanide is going to be made, even presupposing you have all these other things in these special conditions, then it would probably rapidly be killed because of the cyanide and other things, the other toxins that were there. Because it was just a chemical reaction. You don't know where those ingredients came from. And even more importantly, okay, so even if you want to say you made some amino acids, how do they get put together? How do they, propagate? So the next phase of hardness to understand proteins, I mentioned how with that little charm bracelet we had, every one of those amino acids has certain properties, whether it's the size or a little bit of attraction because of charge or repulsion because of same charge. So the protein will fold in a very important and specific way. That would be called the secondary or even the tertiary structure. And many proteins, they have a quaternary structure, which means there's a bunch of different subunits that somehow have to come together to have a specific function. And if they don't come together right, then function doesn't happen. So for example, in oxygen, if you don't have the right things, then you can't carry the oxygen the right way, because the hemoglobin that's in your blood, it binds very tightly to oxygen. It also binds loosely to carbon dioxide. That's when you're sucking air and you're blowing it out, you're blowing the carbon dioxide out because it's gotten there through the blood that's gotten to your lungs that you're now blowing out. And similarly, in reverse, you're getting the oxygen in. And in fact, if there's just even one little change, as there is in sickle cell anemia, there's one substitution of an amino acid, and that just changes the shape of the hemoglobin molecule such that the whole red cell changes shape so that it is susceptible to clotting in small blood vessels and creating all the different problems in the various different organs that sickle cell anemia, for example, just one little change of amino acid. So we can extend the sample calculation. We sort of simply last time said, okay, if we have 100 things and we want to put them together, what's the chance they could come together? And we got this pretty high number of 10 to the minus 58th, even if we gave a 10 to the 150th, but if we had every atom in the universe for 25 billion years at the fastest reaction known to man, we'd still have an almost impossibility. Now, to put that one in perspective, what was the chance that somebody could win the Powerball last night? Something like 1 in 200 million. So that would be like 10 to the 8th or so. 10 to the 8th, I think, comes out. So that means you'd have to win the Powerball like 6 times in a row, 7 or 8 times in a row, in order to have even this chance of 10 to the minus 58th. Remember, minus means it's 1 over 10 times itself 58 times, which with 10 is just 58 zeros. So now, though, we can be a little bit more sophisticated, and we can know that, well, proteins, okay? A small protein, most of them are about 400 amino acids, but if we just take a 100 amino acid, and we want to choose from those 20 acids, and remembering that's got to be the right chirility, so it's a 1 in 40 chance 100 times over, 10 to the minus 170th. It's just an unfathomably low number, a low chance. And again, to put that in a little perspective, the beta-lactamase, which is what bacteria have to be fighting off or be resistant to penicillin from, and penicillin's a problem now at the border, right? But anyway, it's a 10 to the minus 245 chance. And if you compare, the number of photons produced in 20 billion years by the sun is only 10 to the 65th. So again, these numbers, they just throw those numbers out. Unless you kind of have a little concept for numbers, you can just say, oh, well, yeah, why wouldn't it happen? I could win the lottery. I'm going to go, give me 10 for, whatever those numbers are. So, but even if one succeeded and you got this one protein, how would it self-replicate? How would it therefore be available to the next generation to evolve into something more complex? And the overwhelming majority of trials, if it was that way, would be a waste. And every reaction takes some energy. So think of all the energy that would be wasted during doing all this, and we'll talk about that in a bit too. And the assembly itself presupposes the need for some other proteins. So you can't have one protein without another protein to help put it together, a lot of other proteins, in fact. And note, for example, that the simple E. coli makes 4,200 proteins, a little E. coli that can contaminate your church picnic food. Humans make about 100,000 different kinds of proteins. But the interesting thing to me, well, we'll talk about it later, there's 25,000 genes. So that means there's a really lot of efficiency in how they can use the one blueprint to make 25,000 copies of genes to make 100,000. So they make segments that some of them overlap. It's, again, a very complex thing. But it's really much more complex. And I think another analogy we can make is, making proteins, just proteins, is something like a factory floor. So you don't just expect that you put together all the ingredients and they're going to, number one, come together. You've got to have the blueprint for how to put it together. You've got to have the raw materials. You've got to have the assembly plant. You've got to know the orders of how many you need, how many you don't need, regulate that you don't need too many wheels and not enough engines, when you're making a car. So there's just a lot more involved. And then you have to deliver it and package it and get it to your Amazon customer, right? So the blueprint, where is the blueprint? The blueprint is in your DNA, deoxynucleoriboic acid. And the nucleotides are really quite simple when you really get down to it. It's just that there's a lot of them. You've got these amine bases, and there's only 4 of them. It so happens that RNA and DNA switch, but don't get hung up on that. But there's only 4, and 2 of them meet to each other. So there's really only 2 different combinations for these things. And they've got, like the protein I said, there's a backbone, and then there's all these pieces. But then they also meet together like a zipper. So you've got 2 different backbones sticking together, connected by all these little nucleotides. And as I said, they only fit together. There's cytosine and the guanine, the adenosine and the thymine. And they then, because of the size and shapes and energy considerations, they will go into a spiral, a helix. And it was indeed that double helix that Watson and Crick, it's a famous discovery in the 1968 Nobel Prize for discovering the shape, the structure of this. But remember, only right-handed or only left-handed nucleotides can fit. You can't mix them up, and that's going to be only one direction that this thing goes. Now, remember last time we were here, we talked about this Fibonacci sequence. Remember I mentioned that? It's like the sequence is that every succeeding number is the sum of the 2 previous numbers. And I showed lots of examples of how that's just in nature by chance, right? And so happens that the width and the length of one spiral happen to fit a consecutive pair of Fibonacci numbers here. So just saying. Now, the DNA is organized and folded into chromosomes. We all hear about, humans have 46 chromosomes, 23 pairs, and different animals have different numbers. Again, just to give it some, I gave out some wrong information. I got confused about what my one abbreviation was. That man has 3.2 billion base pairs. And there's a pair, so there's 6 billion molecules that are wed together in various of the chromosomes of that DNA. I said there was 6 trillion, but what I meant to say, there was 6 trillion CDs worth of information stored in every gram of DNA. And I told you that if you add up how much grams one person's body has, it exceeds the known storage of, I don't know, it was 1 trillion terabits or something like that of information that's been ever stored in the cloud or wherever that is. And just to put some perspective on that, if you could unravel the DNA in every single cell, it's 2 meters long. It's that narrow, just to give you a sense for the size. And if you added all the cells in your body, it's 10 to the 10th miles long worth of DNA, which is twice the width of our solar system, from Pluto's orbit or Neptune's orbit, And now people will talk about, we got homology. Well, if you've got that many things, even if you're 99.6% alike, that means there's a 12 million base pair difference. So that's not a very effective way of describing how alike things are, because there's not really much room for that scale to understand. Now, you'd say, okay, well, we're the top of the heap in terms of evolution here, so we should have the most DNA. But no, we don't have that. It's not the amount of DNA is not correlating to the complexity, because chrysanthemums have 18 chromosomes, but they can have various other multiples of 9's worth of chromosomes, so they can be variable. The biggest bacteria has actually 25 times the amount of DNA that we have in our body. So, I was going to say, where does all that fit into an evolutionary progression? It doesn't. Now, when I was in college and even medical school, we were taught, only 2% of those 6 trillion 6 billion, sorry, I said it again, 6 billion DNA pairs or molecules, only 2% actually encodes for a protein. And the rest of it, which we call exons, and the rest of it is what's called introns. And they were called junk DNA. And it was portrayed, and may still be in certain books when you pick them up and read, that that's left over from evolution. That's just the stuff. But as it turns out, in the last few decades, we've learned that those are very important in terms of these introns are very important to help us have regulation, to tell the gene when to turn on, when to turn off, when something else is needed, and things like that. So they're very important regulatory elements. And there's these start and stop sequences, all kinds of things. And this is what I was saying earlier. 100,000 human proteins are produced with what seems to be only about 22,000 or 25,000 genes. So there's a lot of efficiency in how that's stored. So now that we've talked about what a protein is and some of its structure, we talked about how it's made. The first stage is how do you get from that DNA to the RNA? So the blueprint has to be first transcribed, and that's where it's transcribed by splitting that zipper apart and taking a template of RNA from it. In fact, it's specifically called messenger RNA. And that happens actually 300,000 base pairs per second. It's really quick that it can do that in the cell's nucleus. So this is this complex molecule that's having a very fast reaction. And the process, though, if you can imagine, it requires that DNA has to unfold, it has to unzipper, and only parts of it at a time will unzipper to allow that DNA to duplicate the one strand. And it requires a very complex protein called RNA polymerase. And just in order to make a protein, as I was saying, you need this 10-part protein itself with many other regulators and proteins in order just to make the protein, to transcribe the RNA from the DNA. So that's the first step of making the protein. So it has to bind in a very specific way, and because of the way it's folded, it's got an active site, and it can do all the things that it needs to do. And by the way, DNA proofreads the mistakes. There's a part of it, there's other molecules that proofreads when DNA is made over. And in fact, I think I'll mention it again, but that's why RNA doesn't have that. And most viruses are RNA, right? And that's why viruses can mutate much quicker. They talked about, the COVID virus mutating and changing. Well, because it doesn't have the proofreading mechanism that humans or regular life has, eukaryotic cells have. So, because viruses are not life, right? So now, once you have the RNA, which was in the nucleus, it's got to somehow get out of the nucleus and into what's called the endoplasmic reticulum and find its way to a ribosome. In the process, it has to go through some further splicing and packaging and cutting out some of those introns. And it has to then find a so-called transfer RNA molecule that will then usher or escort the amino acid to the growing chain of polypeptides that will ultimately then therefore become the protein. And this is where the code really is. As I was telling you that there's these pairs. So every 3 of these pairs specifies an amino acid. There's a little overlap. Some do 2, some do 1, some do none. But if you can make it, if there's a four choices here times 4 times 4, so there's 64 choices you can get out of a 3 position, 4 options. So there's about half of them. Most of them have a duplicate. So about half of them will be present to do that. So, and that has to take place on this ribosome, but more on that later. That ribosome, here it's later, the ribosome itself is a 2-part, small and large subunit, a little difference between the prokaryotic cells and the eukaryotic cells, which we'll talk about that in a minute as well if you don't remember that. And this is, look how many proteins are already involved in the structure of the ribosome just to make a piddly little protein. So there's already a lot of proteins to make a protein at every step of the process of making that protein. The codon is actually RNA at this point, but you could speak of the DNA. Pardon?
Those little machines.
The codon is just the 3 prongs that specify that we're going to talk in just a moment. Actually, that the transfer RNA will then usher. It's carrying one of these amino acids, and it has to recognize those 3, and it will deliver the proper one. Is that the question?
The ribosomes and all those special little machines are also coded on the DNA. So you have the DNA to make the ribosome.
That's true as well. Now, some of it is made actually on the ribosome makes some of its own RNA, but so it gets complicated when it's beyond the scope of what I know well. But you're essentially right. So you already have to have DNA encoding what's already got the workbench that's already there to make the product. So even if you got that most nascent, earliest, most simplest protein, it needed all this other stuff in order to be made. So, and these are ribosomes and electron microscope. And I was trying to make this very point. That you needed to have a lot of infrastructure before you can make one product. Now, the transfer RNA is what I was just referring to. That's what I thought your question was. And that is actually also encoded in the regular DNA. So every single amino acid has a unique specific transfer RNA that will be that chaperone, if you will, that brings it in. It itself is complex. It's got up to 90 nucleotides. It's folded in just the right way, so it will nestle into the ribosome the right way and deliver its component just the right way. And there's many other parts that help that to happen. And by the way, if you kept track of how it was copied and then how it's going to be made from, it's got to go in the opposite direction that it was made in order for it to be equal to what we started with. So again, that's something. And then there's a lot of things that happen at this translation, is what it's called, of the RNA into the protein. A lot of other processes right there in the end result at the ribosome. So now that we understand that the blueprint is the DNA, we can understand better what is a point mutation. You must have heard of point mutation. And when any of these things mutate, it's because one little element, or perhaps of the RNA or DNA, has changed or has been deleted or has inverted in some way, shape, or form. And you have all kinds of terminology for whether it's the sense side or the non-sense side, and again, beyond what we want to get into. But there's all kinds of ways that can happen. So the mutations have to be at this nucleotide level, and we have 3 billion of them. And if you want to take the other side, you can say, well, okay, so what if one or two of these things changes? You got 6 billion other ones. So it's going to take a lot of change in order to have a good end result. So they can't have it both ways. It can't change gradually when you need to change so much. And we're going to throw a number on that, and put a number on that from Spettner actually in just a little while. And I mentioned how there's a surveillance and repair mechanism. But now, I said last time, it's not a really very efficient way to have mutations be the way that you build information, because they're usually harmful and usually lethal, and they're almost always harmful if they have anything at all. There's 1.6 harmful mutations per generation. I think I might have pointed out last week how there's this article in Nature. It's not a right-wing publication that shows that we should be extinct if there's that rate of mutations over the course of millions of years. And by the way, it's only the germ cells mutations that are going to have any potential to pass that on. So if you get a change in your somatic somewhere else in your skin, that's maybe cancer or a mole or a growth or something that's not going to be passed on to the next generation. And scientists have been studying the Drosophila, the fruit fly, for decades. And there's 20,000 mutations in that fruit fly. And it's still a fruit fly. It hasn't developed into a different species. And there's never been an example of making a mutation that changes and can demonstrate from one species to another. And so I talked about this as cancer. There's many, if not most, if not all, mutations actually don't represent new material. They represent an uncovering. If you can imagine, if one of those nucleotides drops out, then that means the code shifts a little bit, so that's going to change the output of that code. Maybe even change the stop sequence so that it won't make so many. So it's not a very efficient way to work, is the bottom line. So I dwelt on proteins here in quite a lot of detail just to give you a sense for the depth of even that one area. But we have carbohydrates and fats, not just for for the health industry, but we need them for certain things. How else would we fill out our, but anyway. And then there's amalgams with, mixtures of lipids and carbohydrates to do all kinds of really important things, signaling and keeping the maintenance of the functions of the individual cell. And each of them has a process to regulate them, break them down, to store them, to unstore them. But there's one thing that I haven't mentioned yet that's critical to any good factory. Energy, right? You need to have energy to run all of this. So if we're going to store, and we're going to do all these chemical reactions, usually chemical reactions are going to happen downhill, right? So you need to have energy to put into, or uphill rather, so you need to put energy in order to make that happen. And I'm not going to go through this, don't worry. We're going to focus on the fact that the final common pathway for most energy in everything is the citric acid cycle you may have heard of, the so-called Krebs cycle. It also is called the TCA, a tricyclic acid cycle. And it's in the mitochondria, and it is innately, it feeds the so-called electron transport chain that we're going to talk about just a little bit. But there's all these very complex means of breaking down proteins, carbohydrates, fats, and other things that ultimately lead to getting the energy in the form of the ATP that even the smallest of cells use. And this is it. But now let's make a little bit of a pass through this and say that this citric acid cycle itself requires 10 enzymes with an average of 330 amino acids, including subunits. And the net result is how much ATP we get. But look, if we just calculate this all out, the chances of our just making those 10 enzymes in the right order is 10 to the 818th, just numerically. Okay? And then it's got to, and that's only just to get those things the input, the glucose, broken down and put into the cycle. Then all these oxidatable things get into the electron transport. And the thing is, even the simplest of cells, the single-celled organisms we have today, use ATP to get the energy. So again, it's not like we went from wood to coal to gasoline to nuclear here. Okay. So, and this is only happens on the mitochondrial membrane. There's four key components to this, and if we just say the first subunit, the first one has 44 subunits of proteins that go together. The others have less, but the point is 4 of them have multiple subunits just to make the energy to make the whole process run. And there's multiple cofactors that are kind of enzymatically help these things go. And you might see in Costco, your CoQ10 and things. That's actually supposed to help you make your Krebs cycle work, but it's hard to imagine it gets into that little mitochondrial membrane just the right way, but I don't know, I'm not. But this is the key. I mentioned this one earlier, this ATP synthetase. It's in every single cell that's ever been discovered and known to mankind, and it's a complex 10-unit. I think I have 10 units of proteins mixed together, and that's from the very start, and it had to be that way from the start. Now you'd think as well, okay, if we are consuming all this oxygen over evolution, wouldn't we run out of oxygen and suffocate and nothing would live? Well, that's right, we'd be making carbon dioxide. But the beauty of it all is that plants take the carbon dioxide and turn it into oxygen through a similarly complex process of moving electrons around and building up and making the ATP so that they can function. So it's the ultimate in green energy, okay, literally. So, and it's the all these complex systems that if they weren't working from the day one, the species would die. So to summarize here, our first segment, the biochemistry, organic chemistry part, we had to have complex molecules had to first appear, at least some of their components, such as the amino acids, the carbohydrates, the lipid complexes. There had to be a blueprint that appeared somewhere or was designed, to direct the formation of the proteins and the regulatory stuff that goes with all of that. And there had to be a way of saving and transmitting that information, which we're going to cover in the cell in the next thing. And there has to be really complex yet effective control mechanisms. Like I say, you can't just keep making tires if you're getting ahead of how many engines you've got, or else you're not going to have an efficient factory floor, right? And they all had to appear with a great deal of complexity from the start. And you had to have an energy system that, by the way, is a universal energy system that has not in any way shown signs of evolution from the simplest to the most complex living that we have today. And the settings to protect, and not to mention, once you've made your product, you've got to wrap it up, and we didn't go through all that. We'll talk a little bit about it in the cell, but not much. It has to get out of the cell to do whatever it is that it needs to do, So, and they're just from the simplest to the most complex has it, and we don't seem to see any leftover or failed products. I mean, if we made, if we had a bad die for the bolt that holds the engine to the frame or something, we'd have a big pile of waste, right? And we'd have to melt it down and use it again. But we don't have all those. We should have just much more waste than we do of product, and we don't. So, and it all had to work from the get-go. So let's just revise these properties because we're becoming increasingly sophisticated here, aren't we? We talked about the individual components, whatever they were, and we talked about proteins. But now we can say, well, if there's 60,000 proteins, and we said there's probably more than that, but because there's a little overlap, we just be gentle, 60,000 of 100 different amino acids, and there's an average is more like 3 times that. And you had to have all the right proper ingredients and circumstances and systems to control all this. We'd find there's 10 to the 4 million and something combinations. And if we then wanted to even get more detailed and know that that had to be based on what the nucleic acid codon options are, it'd be like 10 to the 24 million. And this, I didn't just dream this. This is this Spettner guy that I was just referring to. This is a mainstream kind of person, a biochemist that came up with that number. And this is how he came up with it, figuring that there's of a 1%, he just used 1% of the 6 million billion base pairs, you could get that much variation. So, boy, I think Las Vegas would love to give odds on a number like that, but, So how could such complexity and precision be anything? I don't know. How could it be anything but formed from knowing the hand of a knowing creator? So next we're going to go talking about cells, because cells is really the link to life, right? Every bit of life has a cell, and a cell is very complex. So we've covered this much here. Now we're going to try to cover things about what's called abiogenesis. How did a cell first form from non-living material? How did something living like a cell first form? We're going to talk a little bit about that. We're going to just highlight some others of the organelles, the components of the cells, and very importantly, how the cell reproduces, how the group of cells that are, a being reproduces, such as a human being's on a level, something about genetics as well we'll talk about. And so the purpose of this section really will be to highlight how complex and multifaceted cell function is, the structures, the processes, and how it reproduces itself. And then we're going to try to extend it to the organism level, where we'll talk more about chromosomes and mitosis, meiosis, growth modulation, things that you've probably heard of, but we're going to try to put it together here. And again, we'll see many more examples of how things are really much more complex than the cute little cartoons that you might hear somebody with a hand-waving explanation explain how this could have evolved to that, could have evolved to that, could have evolved to that. And we just, again, to appreciate throughout, as all of these lessons, the monumental steps necessary to make even this one of 10 or so steps that we're going to go over to make, man out of nothing, and just, I think, impress upon us what's the utter impossibility of a step-by-step process to cause this to happen. And I thought it was interesting. I came across some reading at one point. You remember when Pharaoh had his magicians at the 10 plagues, at the time of the 10 plagues. The first plague, I think he turned the water red, but his magicians were able to do that. And the second one, I think he made all of the dust to turn into frogs, I think it was. And the third one was to create life out of lice. And that's when the magician said, no, we can't do this. So I said, only God could do this. Okay, so even from back in Exodus, we get a hint that only God could make life. And that's kind of an interesting little concept. So life from non-life is just a brief history of thought. Aristotle he weighed on just about everything, didn't he? But he said, it's just spontaneous generation from things that are just putrefying and smelly, and then all of a sudden they can start growing again. Well, okay, he didn't have much to work with. Francisco Redi in the 17th century actually was the one who first showed that maggots, that rotting flesh, does not result in new life. And he concluded that life was beginning by the order of supreme and omnipotent creator. Again, they didn't have Nobel Prizes in 1668, but he would have gotten one for that. Robert Hooke, who's the one who discovered really the microscope, the cell, by using the microscope, Leeuwenhoek the microscope, but he discovered the cell and published about it in this book here. So he came up with the idea that things are made of cells, because before that we had no idea. They couldn't look at a small enough level to know that. Yet he still thought that life was coming from, he saw moldy leather, and he figured, well, that life is coming from something that's breaking down. Erasmus Darwin, who was the grandfather of Charles Darwin, posthumously, his book here was published, or his work was published, and he said that there's no absurdity in believing that life is produced by decomposing organic matter. okay? The French Royal Academy, I think I might have mentioned this the first time, offered a prize to anybody who could disprove spontaneous generation. Well, Louis Pasteur did, so he must have gotten that prize. But yet, Francis Crick, the one I mentioned, who with Watson and Maurice figured out the double helix structure, and NASA strongly still support the concept of abiogenesis. More on that after we learn a little. Now here's just a blatant example of bias. NASA's definition of life, this is their quote unquote, life is a self-sustaining chemical system capable of Darwinian evolution. Okay, so here's an example of starting with a conclusion and working towards the conclusion. And we see that not just in science, right? We see that in law and politics and everything, right? But, so it's very biased from the very start. And I actually put the link in there. But so life from non-life might be the biggest hurdle of all for evolution. I mean, I've given you some pretty big ones with 10 to the 24 millionth, you know. But so I'm sorry. Was there a question? I thought I heard somebody. I don't mind taking questions, little questions, but we don't want to get too off the track. But there's certainly will stay till whoever wants to ask any question, I wouldn't leave at the end. So if I can get the answer, but I mentioned these terms prokaryotic and eukaryotic. A prokaryotic cell is different in shape. It's got some different. It's very complex, but it's different from a eukaryotic in several features. And that's those single-celled organisms like bacteria. You know, you must have done the high school, looking at the amoeba under the microscope, or the euglena, the flagella, and all that kind of stuff. But the eukaryotics are like plants and all others, you know, obviously including us, but even right down to like fungi and things, because they can put cells together to make a shape and a structure of something. But as I say, even the simple prokaryote already used ATP, it has proteins, it has RNA, DNA in it, not RNA, DNA in it. So we talked a little, now cell structure, I mentioned the nucleus. I think any of us who've looked in the microscope, you know what the nucleus is. Again, the nucleus is where the DNA is stored, among other things. The nucleolus is really within it, which is where they're stored. And we've talked about some of these other, the ribosome is really the workbench, the assembly line where the protein is made. We didn't talk about, I mentioned the packaging plant, the powerhouse, the mitochondria is really where that ATP is made and stored to run the whole process. But there's all kinds of other things. And one that I want to show here is this so-called ground substance. What is it that's between the cell wall and the nucleus? Okay, I got to point it that way. There's one more thing I want to talk about before that, I guess, sorry. A truly amazing feature, and I made reference to this before, is that even the single-cell organism, one example of how there was already complexity, is that there are these microtubules that get put together. And these microtubules then have some proteins that make those microtubules sway and move, and that's how single-celled organisms move. Because another requirement for life is that it has to be able to move, to ambulate, to get from one place to another. So, and as it turns out, there's a very definite structure and reproducible. We've got that same structure in the cilia of our respiratory epithelium. That's how you must have, if you take something in, some of that Canadian smoke or something, or cigarette smoke or something, it would just get right down in your lungs and really gum things up, but there's a constant flushing mechanism because of the cilia of the epithelium lining of your lower passage, your upper really passages, that is constantly pushing that stuff out. And that's why the smoker's hack, they're always going to cough up a bunch of stuff because their cilia are still trying to flush out that bad stuff. So a thing that I think was really, really amazing, again, when I was training and learning, the first thing is that we just called this ground substance. We didn't really know, they didn't teach us. They were beginning to think, well, yes, we've got certain things that help the cell to move in there, the actin, myosin, but as it turns out, there's a superhighway of microtubules in our cells made of these microtubules. And the microtubules are kind of the area that what do you call it, when the grabbing on places, when that rock climbing kind of thing, for proteins that can then carry things, and just from a little bit of a chemistry change, I think it's just a hydrogen, it changes the shape and it makes it walk literally right down these superhighways of microtubules. Every single one of the cells in your body has that. I mean, and again, it has to be an all or nothing. So that's how the product can get at least to the front door to get out of the cell, among other things, because it goes both directions too. I think I even say it right here that they understand a one class of proteins carries things away from the nucleus, another class of proteins carries them towards the nucleus. And there's cell tension. This is what they knew when I was studying these things. But what's really unique about life, not only is the cell, but that the cell can divide. It can reproduce itself. And in order to do that, the first thing, it's got to be able to replicate its DNA. We talked about the DNA. That was that zipper. Remember, that's got the base pairs that code everything. And even the simplest cells use DNA. They have a plasma membrane, not a cell wall, and they don't have a nucleus, so all the DNA material is kind of floating around or grouped together in what's called chromatin in the cell. But all the rest of multicellular organisms are called eukaryotic, and they have the cell membrane, the nucleus and all. So, and the simplest matter of division is mitosis, and that's when a cell just splits. And a cell will just split, and it will split with an identical copy of what it was. So you have 2 identical twins, and then 4 identical twins, and 8 identical twins. But what really, and yet, even with this, there is room for some, we'll talk about it a little bit, there's some room for some mixing of the genes. So that's how come there can be, one rose bush has one color, and you cross it with a different rose bush, and you get a different color. And mitosis is something like our cells will do if we have a cut, wound healing, if we're just growing, what have you. Meiosis, though, is what really first reduces the chromosomes, and it's in the gametes that and then they reorganize and come together. So one of the 23 from the mother, one of the 23 from the father, and then they mix in just the right way, and that's what gives rise to all the variation, how one kid can look very different from another kid, yet you can tell that they're probably from the same parents, right? Again, without going into any details. So let's just get into how does the DNA itself replicate? Well, I indicated that chromosomes in the human, there's 46 chromosomes, 23 pairs. All the DNA is grouped into those. But the DNA, I told you, it's 2 meters long in every single cell. So it's got to be rolled up and kept, and there's all kinds of proteins called histones that help fold this, and they have to become unfolded in order for this to happen. So just think of some of the upper level, the high level, just not in the details. What has to happen? There has to be a signal to change, to duplicate. There has to be an unraveling, topoisomerases. There has to be some cleaving of the DNA molecule. And this happens simultaneously, both in 2 directions, both sides at the same point. There's a proofreading mechanism as it's getting duplicated, as this zipper's coming out, and there's a new zipper mate that's being added to each one of them. And then it has to refold and recoil, and yet that happens really fast. 1,000 nucleotides per second get copied in this mechanism. So your entire genome, because it can be working at different levels, can get copied. Those 6 billion can be copied in 40 minutes. And I thought this, I just conceived of this idea. Copying our 6 billion base pairs is like taking 400 skeins of yarn and having them all bundled up and having them somehow get duplicated in 40 minutes, 1,000 nucleotides a second. So if you've ever, I remember my mom used to have to, it would be a real mess when she was a knitter to get that yarn, from being tangled. If you can, 400 skeins of yarn all jumbled up and then you're pulling it apart, Heck, we complain if that's one length of an extension cord, right? So anyway, so the principal protein a protein structure complex that does this is also bacterial. This is just a bacterial. It's called a replosome. It's 10 proteins that are melded together, and they form this like ring, and along the ring is some other components that help all these things to happen. So it's kind of like pulling a string through a ring and having 2 strings come out. That's. And again, without going through the detail. Now, Jacques Monod, who was a famous, and again, another Nobel laureate for his understanding that there's a genetic control of different enzymes and how viruses were synthesized by E. coli, this concept of a working unit, an operon, he's the one who said, we have no idea. The primitive cell might have been the simplest, was the same as other living beings with regard to the genetic code, like human cells. Thus, the simplest cells have nothing primitive about them. So he was honest about it. Yet, Thomas Huxley, Darwin's bulldog, he says, I find no record of the commencement of life, but, ever faithful, I should expect to be witness of the evolution of living protoplasm from non-living matter. Okay, again, you have a bias you start with, you're going to be stuck in that bias. Now, once the cell has the chromosomes line up, there's a very well-designed set of, or a very well-defined set of steps that they line up, they split, they go, and then there's cleavage, the cells, the cytokinesis. I don't want to get into details again as much as to say that just the division, the physical division of the cell, once you've got everything duplicated and all ready to duplicate, is also a very complex and well-regulated set of steps. So, with complex regulation. Now, many of you might have heard of Mendel, who's the one who's considered the father of genetics. And he was, maybe I got too close, is that way? He was a friar, an abbot somewhere in Moravia, somewhere in the mid-1800s. So he was a contemporary with Darwin, but Darwin knew nothing about his studies because if he published it in something very, very obscure that was rediscovered actually after he died. But yet his principles, he did these studies with pea plants and understood that when you cross one with another, he's the one who figured out that there's different alleles, different parts of a chromosome that can cross over and add into another one when they're dividing, and really the concept of a gene. So Darwin, when he was making his decisions, his theories, nor did his contemporaries know anything about the beginning of genetics, and it's this genetics. In fact, when I was telling you about how, the DNA, so you might call that molecular genetics, in fact, because we can define genetics down to a molecular level. And molecular genetics didn't really start until, the 1960s, 1970s, and it's been rapid in the last couple decades. But even 100 years ago, and 150 for him, they didn't know about what Mendel figured out. And it may well be that some of this crossing and things, that might be how come Jacob got all his spotted sheep. But I've read something on this, but it's more complex than this. But they don't have it 100% figured out how he, But anyway, pardon?
That's exactly what he did.
Well, that's what he did, but somehow we don't understand the genetics well enough to know exactly how that all worked out. But but that's an application. So I'm trying to bring it home. We are in a church. So anyway. So then it's much more complex, though, with what's called sexual reproduction, when you have the different haploid pairs coming together. And notice that, and this is a simpler thing here. This was for flowers, but there's an end of every chromosome that's called a telomere that is kind of a capstone protection. So it won't get dinged, so the rest of the real guts of the chromosome won't get dinged up by whatever movements and reactions there are. I'm going to come back to that. So that's an important thing that's going to factor into humans in a bit. But again, we have this, and that's how different. It's much more complex. There's all these other things: linkage, crossing over, pleiotropy, polygene. All kinds of extra things can happen to make variations from one person to another. But none of that changes a species. And again, we don't have to go through the details. It's just as much to know that there's yet another thing you never really think of that's very, very complex, yet ordered. Now, meiosis is when I told you we have haploid, means a half number of the 23 chromosomes in humans. So you can't propagate unless you're mating two species with the same number of chromosomes. So dogs, for example, all dogs have 78 chromosomes. Humans have 46. If you cross, I mentioned this, I think, a couple weeks ago, a donkey and a horse, you can get a mule, but you may know that the mule, they've got different numbers of chromosomes that the horse and the donkey do. But the mule is then sterile. It can't reproduce because it's got to have the right number of chromosomes. All cats have the same, I think, is it 38 or 78 chromosomes? I'm not sure which. And that's why if you've gone to the zoos, they evidently only exist in zoos, you can get your ligers and your tigrins, a tiger and a lion can mate. But it turns out there's other things that they'll be less fertile and they have a lot of other problems, so they won't propagate because it's an artificial to get them to propagate in that way. So, but the point is that your house cat could mate with a lion. I don't want to think about that, but because of the right number of chromosomes. And that's why you might extend that to say that there's different kinds, right, on the arc. So you could have all cats could have led to all cats we have now. All dogs could have led to all dogs we have now, because at least it's possible because they have the same number of chromosomes. So, but it's not possible to have, other things propagate. So.
And that's one of the big arguments is how could Noah have gotten all those animals
Right.
on the ark? He didn't need them all.
Yeah, and we're going to cover that in about 4 more sessions. Yeah. And that's the concept of kinds versus species. And the definition of species has changed mightily over the years. And Darwin only had a certain number of species, and he was basing it completely on what it looked like, the so-called phenotype. And that's why he was able to, fill in interpolate a lot of things that aren't supported, for example, by some of the things now on a molecular level we know to be the case. So, right, great apes. Now, in fairness, to try to present the other side, evolutionary theory says, well, the way great apes, which have 48, so a human can't mate with a gorilla because there's a different number of chromosomes, is that there's a chimp chromosomes that fuse together to make one, and that happened in both sets. So that's what they say. I don't know, but against the theory of this, remember I told you about those telomeres, those caps that were protecting the enzymes. The telomeres, they're 6 base repeating sequences, but there's like 15,000 of these at the end of every single human chromosome, and chimps only have 800. So it doesn't make sense that something as well protected as a human chromosome could possibly have developed from something that was less protected. So, the human chromosome is very protected, like the White House. The chimp is less protected, like the public library or something. Just an analogy. No politics here. And furthermore, a fusion would leave that chromosome very degenerate, and we don't see this sort of remnant signature of such an event. There's other ways you can look for pieces that maybe the introns or other things that don't have anything. So it also just doesn't make any sense it doesn't make a lot of sense that would happen. There's just a shred of possibility that's been built upon to make a bigger theory. I mentioned this before, mutations are as adding information. And that's kind of counterintuitive, and there's never been a definite example of showing that a mutation has increased the complexity of something that wasn't an uncovering of what was already there. Something that was in the code that then, as I say, we lost or inverted, flipped over, or somehow got added to, that there was a little modification of the code of information that was already there, that just got uncovered because of that mutation. Anything positive has been known that's how bacteria. There are certain examples of bacteria that will be resistant to one antibiotic, and then after a while, they'll flip back and be resistant to another antibiotic because the same information is there. They've just reinverted the information that was in their DNA. And there's many, many, many examples. This was the example I was just thinking of, the resistance to streptomycin. There's one little nucleotide change in the ribosome only that makes this change. And other bacteria that supposedly can learn to grow on certain media but not others, it's also an example, and there's been shown examples of a point mutation, and then that point mutation can invert back, and they can't grow. So there's just all kinds of complex controls that I think even the cell biologists would agree they've only scratched the surface on understanding. So progress really would seem to be, of the mutation, would be by losing more information. How could you go uphill? So now, to try to just give a very quick run through, what happens when there is fusion? So those two gametes come together. Well, after about 90 minutes, there's another division, and then it enlarges through this multiple and very well reproducibly, well-controlled set of processes through different things. They're called a morula, a blastula, a gastrula, and I get the wrong one again. And in a very orderly way. And we talk about, once this gets to be in this size, the germ cell layers. There's an ectoderm. The ectoderm is what makes your skin, and it so happens that because your spinal cord is external first, all your nervous system is ectoderm. And when you talk about a carcinoma, an epithelium. A carcinoma is an epithelial, an ectoderm-based cell. A sarcoma is a mesoderm-based cell. It's something that's inside, like your bones, and I might be wrong about that, but your bones, and then endoderm is some of your internal organisms. So that's why you don't get a carcinoma of your well, I don't know. I don't want to put out bad information, but the point is that there's germ layers, and that's how sarcomas and carcinomas are named, because of the germ layer, which is to say, the layer on the primitive developing embryo. So, and yet so it goes through all this, and a baby comes out. Okay, so this is not just like, stir it for 15 minutes and reduce it to a simmer and you've got a baby. There's unbelievably complex modulation factors you can well imagine that control what folds where and what develops when, and the umbilical cord goes here and the heart, all these different things. And each one of these cells, and here's a good question for you, how different do you think like the DNA in a heart cell is from the DNA in a skin cell? None, no difference whatsoever. It's all in how that is, this is a smart group, I guess. I got to get tougher questions, but it's how it's expressed. And we only have, again, a rudimentary understanding of some of the factors that control how one thing is expressed versus another. So, and that's the whole promise, right, of stem cells, because stem cells ideally are less differentiated, so maybe you could make them or put them somewhere else and influence them under the influence, of course, of what's already influencing cells there to make them develop into something else. But, they're knocking on the door of that, but we're not there. But again, just imagine all of the sensitive control and regulatory structures from the beginning of all of this, the genetics of it, all the way through the development of the embryo. But back to the cell. Once it's duplicated, it has to grow. And there's very well-defined cycles, cell cycles, growth cycles, synthesis of the DNA, another growth cycle, and preparation for division. That each one of these is a very, very complex set of regulator cells, regulator substances that control how much and how long and when it all happens. And a lot of things can go wrong at this stage. For example, basal cell carcinoma. If things go wrong, then you've got a problem. So usually when things mutate or change, it's for the worse, not for the better, right? In fact, it's almost always. But so again, it's not a very efficient way to expand or evolve to have these kinds of mutations. This is something that's only really been, well, I guess it was first the concept of it, but it's only been better understood in the last 20 years or so, something called maybe 40 or 50, but time flies, but apoptosis, which is to say that every cell in your body, well, not every, but many cells in your body, they live for a certain period of time and then they die, and then they get replaced by other cells. Unfortunately, as we go along, we lose more cells than we replace, but the concept of when a cell dies is called apoptosis. And I think, I say it here, yeah, can you imagine this? I just think this is a very literally mind-blowing thing, is that in the normal development, half of your brain cells die during gestation. But somehow it's the right ones that you didn't need, or it's the duplicate ones. It's not like you lose one segment of your brain and therefore can't walk or something. Somehow there's processes that know how to weed out and reduce the number of cells in your brain by a factor of 50%. And in the eye, the nerve, and we're going to have a whole section on the nerve because I'm on the eye, but the optic nerve gets reduced by to 1/3 or even 1/4 of what originally was. Yet you don't have normally blind spots all over or lack of function. So it's just an amazing process. Now, this has been seized upon. Why? Because it's this apoptosis that is involved in the aging process. So if we could somehow figure out how to regulate, modulate the apoptosis, well, we might have eternal life. There's an easier way, but we might be able to have, this is how many cells you lose each day. But anyway. And again, it's going to be very highly regulated. There's been Nobel Prizes given out for understanding the genes and the signaling and the process that goes into this. And this is just some beginning understanding of how maybe they can modulate this cell, the cell death in the prostate cancer model. So abiogenesis is kind of what we're setting the stage a little bit for by going backwards. And that's the beginning of a cell, the beginning of life from non-life. And that's just an unbelievably huge step. And there's a bunch of people who thought earlier in the earliest 20th century that, the cell is just kind of aggregated together, the fats came together and made cell membranes, and other things happened. And I told you about how the amino acids maybe could have been formed by this artificial chemical reaction that Miller and Urey did. And I thought it was 52, but I got 53 there. And then others have done things that they think are important and maybe proved that you can make cells from nothing. This guy in 65 is self-replicating proteinoid microspheres. Well, he put amino acids in there, and he had all kinds of special circumstances that made some cells duplicate, or made some stuff duplicate. There's this other thing I don't have on here called ribozymes. That's a big deal. And the ribozymes are thinking that, well, maybe there was originally just RNA, and they can show small segments of RNA that if they can adhere to clays, it's called, but that's not the kind of clay you see on the roadside. It's like minerals, that maybe that can somehow make them react. But again, they really have almost nothing going. And I thought that was a good one from Salt and Pepper. But anyway, so Rudolf Virchow is called the father of pathology. And from 100 plus years ago, he says all cells come from cells, and that's a dictum that hasn't been disproven. The closest anyone has come is this guy Craig Venter, who led a study that showed that they made this particular organism called a synthia, but they really didn't make the organism from nothing. They mixed together some DNA from an E. coli and from a mycobacteria. And they made very special conditions. And I mean, it's a real cell biological tour de force that they did this, don't get me wrong. But they didn't start from nothing. They started from other things that were already cells to get there. Just like, doesn't it sound similar to what we talked about? In order to make proteins, you already have to have proteins, right? In order to make DNA, you already had to have DNA. So he's more famous perhaps for this American Genome Project, where once he made his name in academia, he started his own company or two or three, and that's how now it used to be impossible really to sequence our DNA, our genome, but now it can be done pretty easily, although they're not sequencing the whole genome, and that's why that 23andMe really isn't your genome. It's just a few high spots, so to speak. But anyway, a lot of stuff's been written. Another book that I thought was really interesting is this, I read relatively recently, is this Tan and Stadler book. And he goes through in great detail, these are cell biologists as well. These aren't just people making up ideas, about how many complex things have to happen in order for a cell to actually come together and be a cell. And every one of those steps is far from ever having occurred. I lifted one quote out of there. So it's like asking rocks to organize themselves into a city. And it's probably more remote than that when you get down to it. But now, some honest biologists, I quoted Werner Arber earlier. He's the one who I told he got a Nobel Prize for restriction enzymes, but about 1982 or something like that, 78, I don't know. And he says, you can read it, I must confess, I don't understand how life came about, but because the most primitive cells may require at least several hundred different specific biological macromolecules. And he says the possibility of the existence or creator of God represents to him the most satisfactory solution to the problem. So, yeah. And here's a couple of evolutionists theories. They've said the macromolecule is a jump of fantastic dimensions which lies beyond the range of testable hypotheses, and it's just conjecture. And they were being honest. They want to believe evolution, but they're agreeing that it's just too much of a stretch. So to summarize then this second part about the cell biology, my goal here has been to witness to you the complexities of just coding of DNA, and then consider the complexities of it duplicating and being preserved for future generations, knowing that everything requires the right conditions, the right setting, all these different organelles, the right ingredients, and all these different conditions, and it has to be housed in a cell, which itself we've just gone over is unbelievably complex. The cells need to grow, and they're under great regulation to how they grow and how they differentiate what cells they become, and how they get protected, how they die in the right time so they're not creating new problems, and how this is already intact in the simplest of cells and organisms. And all the while, any of the things that might go wrong would not add or help, but it would kill the species. So here's the age-old question. I thought this was cute. Anyway. Chicken or the egg? Well, we need both. They both were from the start. You can't answer the chicken and egg question because it's, and that's what that is. So, again, I thank you for your attention. If there's any questions, discussion, or contributions, it sounded like there's some people who knew a lot about this.
It seems to me that the process of cells not replacing cells, if you want to, can be the fall of man. Because the time that our cells don't reproduce and we start the aging process was probably about the same age that Adam was when.
Sure. Well, Methuselah is what, 989 years old, was it? Something like that. And
Well,
even Adam was 930, I think, and 910. Yeah. All right. Well, we'll cover some of that actually when we get into one of my segments is on geology, and it's going to include some stuff about the flood. But so we were presumably all vegetarians to start with until the fall, right? So, and then it took a few generations for the negative effects of that to break loose and fall out. Right?
So you can't live with a damaged chromosome, and pass that on.
You can, and that's the problem, right? Some of them you can, and some of them you can't, though. But you can't have a mismatch of them. So.
I had heard that the Black Plague caused the damage to one of the chromosomes.
I just don't know about that one. I don't know.
And it left the ends open like that. And they said that, what is that?
The Black Plague damaging the chromosomes. I don't know. I don't know. People listening online, I don't know if they can hear the question, but So I'm trying to paraphrase a little bit so they'll.
Well, I just wondered if you had any.
I don't know about that one, but I can try to look that one up and look into that. I hadn't heard that one, but of course that was the bacteria carried by the flea that was carried by the rat in the ports. So.
There's a theory out there that the reason lifespans crashed after the flood was because it was a genetic bottleneck.
A genetic bottleneck? Why lifespans crashed after the flood? Because it was a genetic bottleneck.
Well, all the diversity is gone in all the different people. So you have just Noah and his 3 sons and their wives. That's all the genetic diversity you have left. And there's a theory that that's what caused the lifespans to decrease. Because they decrease on a curve.
Yeah, well, then, of course, you could play the story that, well, we had perfection to start with. So so it was just a losing of that perfection, whether it be environmental factors of now having ozone and, who knows what. But there's a number of theories that I've heard in the same vein of what you're saying. But, yeah. So I'm not saying no. I'm just saying, that, again, it's probably more complex than the answer we hear on. So, yeah, question? You're just fixing your hair, or no? Okay, okay. All right. Well, it's 3 weeks from tonight. The next one is, and the next one we're going to talk about will be I call it the march to life. So we're going to see how do we go through the least complex to get to man, and how each one of those steps has an unbelievable set of changes that has to occur, and then we'll illustrate that with number 6, which is the second part of the third session, about all these unique things that I call it other creeping things, some unique features, very complex features amongst things that you'd think should be less complex than us, but like why eagles can see so much better than we can and things like that. But anyway, again, thank you all for being here tonight. Oh.